Hello friends, today we are going to discuss a topic protein. In previous two lectures, we have discussed important things regarding the protein, that is classification of protein and structural levels of protein. Classification helps us to understand the function of protein as well as the nature of the protein, and the structural levels are helpful for determining the structure of the protein. So, in this lecture, we are going to see three important topics, that is types of bonding present in the protein. Then we are going to see another important topic, that is a prosthetic group, and lastly we are going to see important concept, enzymatic action on the protein. So when we consider the structure of proteins, there are different types of the bondings which are helpful in stabilizing the protein structure. So in this we are going to see types of the bonding which are helpful for stabilizing the protein. For sake of convenience, here we have taken protein structure. This, if you look at complex structure of protein, we have studied this at the different structural level, and this protein structure is stabilized by the different type of the bonding. So let's consider the bondings present in the protein molecule. There are four types of the bondings which are mainly responsible for the stabilizing or destabilizing the protein structure. These are hydrogen bonding, then disulfide bonding, then ionic bonding, and lastly we are considering a hydrophobic bonding. These are the bondings which give stability to the structure of protein, and the stability is depending upon the bondings present in the structure. So further moving, we will see one by one how they are formed and how they are going to stabilize the structure. So let's first consider hydrogen bonding for a protein bonding. So hydrogen bonding. It is the most important force that maintains the protein structure. Bonding is shown between a carbonyl oxygen of one amino acid and amino hydrogen of another amino acid, which near at 2.8 angstrom. So this is the bonding which is mainly gives a stabilization to the structure. It is found in both helix and pleated sheet structure. So helix structure and pleated sheet structure, this hydrogen bonding is giving the stability. The hydrogen bonding is found in helix and pleated sheet structure. So we can consider that it is a primary most important stabilizing factor for the protein structure. In helix, it is between the first and the fourth amino acid. The first amino acid, carbonyl oxygen, shows a hydrogen bonding with the fourth amino acid, amino group hydrogen. So it is between the first and the fourth. It is continuing in this manner: first, fourth, then second, fifth, then third, sixth, and so on. So gives an extensive stabilizing of the helical structure. And in sheet structure, it is between the opposite polypeptides. It may in the same direction called as a parallel, and if it is in the opposite direction, then it is called as a anti-parallel. So let's consider structure one by one, and first considering the hydrogen bond. And here you can see a carbonyl oxygen and a hydrogen of the nitrogen is going to show you the hydrogen bonding, and this hydrogen bonding is a stabilizing factor. Here you can see a hydrogen bonding with this. So here is a diagram which shows the hydrogen bonding, how it works. Now let's see in detail how it is helpful in helix and pleated sheet structure to give the stability. So let's consider the hydrogen bonding in a helix structure. Here you can see a hydrogen bonding in a helix structure. It is from first to the fourth. So here we are having the hydrogen bonding. In structural way, you can show this like here. Here you can see hydrogen bonding, extensive hydrogen bonding for the helical structure, and this helical structure is more stabilized or primary factor of stabilization is the hydrogen bonding. Now, in the second, that is a sheeted structure, you can consider here a hydrogen bonding. Now you can see here a hydrogen bonding is there between opposite polypeptides or adjacent polypeptides. So this result extensive network of an hydrogen bonding and gives the stability to the structure. This also we can see by our structural manner. Here you can see the carbonyl and carbonyl oxygen and hydrogen. These two R groups are facing towards the same side. So here we are having next hydrogen bonding. It is from the opposite panel. You can see here one polypeptide chain and another polypeptide chain. So hydrogen bonding is the primary factor which stabilizes the protein structure. Now let's consider the second, that is a disulfide bonding. This is an important bonding between the polypeptide. It may present between a single polypeptide or between the opposite polypeptide, but many times it is observed that it is from the single polypeptide which is resulted by a cysteine amino acid which is involved in the disulfide bonding. The two molecules of cysteines are involved to give the disulfide bonding by the thiol bond. So cysteine is the amino acid which is present in the disulfide bonding, and thiol bonds are formed by the two cysteine molecules or two cysteine amino acid which gives the stability to the structure. Here we are considering the enzyme name or enzyme which gives a formation of a disulfide linkage, that is a thiol disulfide oxidoreductase. Thiol disulfide oxidoreductase which joins the two cysteine amino acids or two cysteine molecules by a thiol linkage which is called as a disulfide bonding. Here you can consider an example of disulfide bonding which is present in a insulin. So in an insulin molecule or insulin, when we undergo for the study, this type of the bonding is found in insulin. So here we have shown with the help of a diagram how that disulfide bonds are present. So this disulfide linkage will give the stability. So this polypeptide chain cannot go apart from each other. Now here we are considering how this disulfide bond is formed. Now here you can see a thiol which is present in the cysteine, thiol which is present in the cysteine. With the help of an enzyme thiol disulfide oxidoreductase, it gives the formation of an disulfide bond. So here we are getting the formation of an disulfide bond which gives a stability. As it is a covalent bond, so it is a stronger and gives the stability. So this is the second type of the bonding which is present in the protein band. Now let's consider the third type of the bonding, that is the ionic bonding. As we are knowing that polypeptide chains are made up of an amino acid, and amino acid have a R group. These R groups are positive and negatively charged. So here we are considering that these amino groups are positive and negatively charged. So we are considering that these amino groups having both the charges. R group may be positively charged or R group may be negatively charged. There are three amino acids which are having a positive charge on the R group, and there are two amino acids which are having a negative charge on the amino acid. The opposite charge R groups give ionic bonding formation. That means ionic bond is formed when opposite charge R groups are coming closer to each other, and this is resulted due to the electrostatic force of attraction. So what is the reason for the ionic bond formation? Electrostatic force of attraction. Two opposite charge molecules coming together and resulted into formation of an bonding, that is called as electrostatic force of attraction. In other words, we are calling it as a ionic bonding. Now here you can see a ionic bonding, carboxylic group, oxygen minus, and here amino group is having a plus. So these two charges get attracted. Similarly here we can consider these two charges get attracted. This is a figure which shows the simple. Now here also we can consider. Suppose this is a one R group with an positive charge. Here is another R group which is having a negative charge. So there will be electrostatic force of attraction between these two, which is called as a ionic bond. So here we are considering it as a ionic bond. Okay. So ionic bond formation is resulted due to the opposite charge R groups which are having either plus charge or minus charge. But if opposite case happens, that is the same charge R groups are coming closer to each other, there will be the repulsion which will destabilize the polypeptide chain or the protein band. So this is the third type of the bonding which is present in the polypeptide chain. Now let's consider last, that is the hydrophobic bonding. It is one of the important or interesting topic. What is hydrophobic bonding? A repulsion by water forces R group together. The R groups which are not positively or negatively charged but having a organic phase, these R groups are repelled by the water molecule. So due to this, what happens? Clustering of an hydrophobic group. Clustering of an hydrophobic group away from the water is resulted, and this gives the formation of an hydrogen bonding, and hydrogen bonding is observed due to the van der Waals forces of attraction. Van der Waals forces of attraction. So clustering of an hydrophobic group. When hydrophobic groups come together, they resulted into the van der Waals forces of attraction. It is simply called as the hydrophobic bonding. Now here it is an interesting picture which shows a hydrophobic bonding. So here you can see these red color groups, they are hydrophobic, or it is a hydrophobic region or R groups, and these are the hydrophilic. The blue ones are hydrophilic, so they are loving the water as hydrophilic, and these are the hating the water. So when it is protein dissolved in a water, or isolated protein will look like this, but when it is in the aqueous solution, what will happen? The outside there will be the all hydrophilic group. Outside there will be the all hydrophilic group. What I will do, I will show it with the different color. Okay. Okay. Now here you can see. First I will rub the previous one, this, and then we will show with the different color. What is different color? Now you can see outside there is a all hydrophilic groups are there, water loving. So water molecule will be use the hydrogen bonding for stabilization of this, and inside there is a combination of all hydrophilic groups. So these hydrophilic groups inside will give the van der Waals forces of attraction. In other words, it gives the formation of a hydrophobic bonding. So it resulted into the more stable structure in the aqueous solution. So water loving molecule, that is hydrophilic, will be outside, and hydrophobic will be inside. So it gives more and more stability. Now here you can also see this structure. Now here you can see this. First understand this is a hydrophobic group. The blue one which is shown, it is a hydrophobic, and the red shown are the water molecules. So what happens? Water molecules when towards the hydrophobic group, the hydrophobic group will show the repulsion, or it will not attracted towards the water. So due to this, what happens? Protein get folded, and it will show a cluster of these all hydrophobic group together. So due to this, what happens? Water molecule is outside, polypeptide bond is there. So this will go for the hydrogen bonding, and inside there is a hydrophobic bonding. So all favorable situations. So it will result into the stabilizing the protein molecule. So these are the four types of the bondings which we have seen in this particular topic, which gives the stability to the protein molecule. Now, if you look at this all type of the bonding in single molecule, how it will look like? So we will show these bondings in this structure. Now, here we can see this is the polypeptide chain which is formed by a peptide bonding. Okay, so polypeptide backbone is there or polypeptide bonding is there. Now in this case you can see here is a hydrogen bonding which is observed here. Okay, so hydrogen bonding gives stabilization. Second, that is a disulfide bonding, it is also giving the stabilization. Now here you can see a hydrophobic interaction, clustering of an hydrophobic groups. Okay, so here you will have hydrophobic interaction or hydrophobic bonding, and here we are considering the ionic bonding. Opposite charge are attracted. So this gives the stability to the protein structure. So here there are four different bondings we have shown which gives the stability for the protein. Now let's move further to the another important small topic, that is the prosthetic group. Okay, in conjugated protein we have seen the definition of a prosthetic group. What is prosthetic group? Proteins have protein as well as non-protein part. Protein having a protein and the non-protein part. The non-protein part is called as a prosthetic group. The non-protein part is called as a prosthetic group, and where we found this prosthetic group, this is particularly present in the conjugated proteins. Okay, so in the conjugated proteins we are considering the prosthetic group. Now what we will consider? We will consider a number of examples for the prosthetic group and what is the group which is a non-protein present in the particular type of the proteins for conjugated proteins. So let's consider a table which derives what are the different prosthetic groups present with the example. So there are number of classes for this, that is a lipoprotein. Prosthetic group is the lipids, and example is a beta-1 lipoprotein of the blood. Then next is a glycoprotein, that is prosthetic group is present as a carbohydrate. Example is immunoglobulin G. Immunoglobulin G. Now next is a phosphoprotein. Phosphoprotein, combination of an phosphate group with a protein. So prosthetic group is a phosphate, and example is a casein of the milk. Then there is another hemoproteins. Hemoprotein heme or iron sulfide is a prosthetic group, and example is a hemoglobin. Then next is a flavoproteins. Flavoprotein, flavin nucleotides. Flavin nucleotides are the prosthetic group, and example is a succinate dehydrogenase, which is one of the important enzymes. Then metalloproteins. Metalloprotein means prosthetic group is a metal with a protein. So there are different type of the ions present, that is iron, zinc, calcium, molybdenum and copper. Iron called as a ferritin, zinc alcohol dehydrogenase, calcium calomethylene, molybdenum, here we are having dinitrogenase, and copper, here we are considering plastocyan. So these are the example of the prosthetic group. You have to remember what is the group present. For example, lipoprotein, so lipids is a group, and example beta lipoprotein of the blood. So that is regarding the prosthetic group. Now another important topic is the enzymatic action on the protein. We are considering the proteolytic enzymes, also called as a proteinase or peptidase. These are the enzymes which break the chain of the protein. So here we are considering the breakdown of the chain of the protein, and a enzyme is called as a proteolytic enzyme. Most of the time it is abbreviated or it is known as a peptidase. So we are considering the enzyme action on the protein. So let's consider the proteolytic enzymes, trypsin and chymotrypsin. Remember the name, trypsin and chymotrypsin. These are the two enzymes which we are going to see. They are very specific in their action. So let's see one by one. First, a trypsin. Trypsin hydrolyzes those peptide bonds. Trypsin hydrolyzes those peptide bonds in which carboxylic group, that is a carboxylic group, carbonyl group, is contributed by lysine and arginine. Lysine and arginine. So when we are having a polypeptide chain, and if the trypsin is going to act on it, then it will only hydrolyze or break those peptide bonds in which carbonyl group is contributed by the lysine and arginine. So only lysine and arginine carbonyl group is present in the peptide linkage. Then trypsin will act on it and it will break down the polypeptide chain. So let's consider the example here. So let's consider a polypeptide chain that is given as a glycine, that is given as a glycine, lysine, alanine, serine, arginine, glycine, aspartate and glycine. As we are knowing that the left hand side having a N terminal and right hand side having a C terminal. So we are knowing the all sign combination. So let's consider what will be the action of an trypsin. So trypsin will hydrolyze those bonds where a carbonyl group is contributed by the lysine. Now you can see here at the right hand side it is giving the breakage which is shown by this arrow. Why not on the left hand side? Because left hand side polypeptide bond, lysine will contribute the amino group and glycine is contributing the carbonyl. While on the right hand side, lysine is contributing the carbonyl and alanine is contributing the amino. So this will break first here. Then secondly, where we will go for the breakage, alanine, serine, arginine. So it will break on the right hand side of the arginine. So it will give the formation of the sequence of an 3 polypeptide, that is glycine, lysine, alanine, serine, arginine, and last glycine, aspartate, and glycine. Okay. So if you look at the structure here, which is same as this polypeptide, so here you can understand why on the right hand side here a carbonyl group is contributed by the lysine. Carbonyl group is contributed by the lysine in this case. So that's why it breaks here, not here, because here amino group is contributing. Similarly, here also arginine is contributing to carbonyl. Therefore, here we are having the breakage. So that is the action of an trypsin. Now let's consider next, that is action of an chymotrypsin. Chymotrypsin hydrolyzes a peptide bond in which a carboxylic group is contributed by a tyrosine, tryptophan and phenylalanine. So we are having a breakdown of those peptide bonds in which tyrosine, tryptophan and phenylalanine is contributing a carboxylic group. Now let's consider an example here. Alanine, lysine, phenylalanine, methionine, proline, phenylalanine, glycine, tyrosine and alanine. Okay, so here we are considering this polypeptide chain. Now let's consider what will be the action of an chymotrypsin. Chymotrypsin, first phenylalanine, at right hand side there will be the breakage. Then we are having again a phenylalanine, so there will be the breakage. And last there is the presence of tyrosine, so here we will have the breakage. So that will be the action. If we have combination of this, that is a chymotrypsin and trypsin. So what will be the action on the polypeptide chain? Let's consider with an example. Now here you can see a series of an amino acid attaches with each other. So chymotrypsin will hydrolyze those bonds in which polypeptide chain or amino acid is tyrosine, tryptophan, phenylalanine and they contribute the carbonyl group. So here breakage on the right hand side of phenylalanine and a tryptophan. Similarly, what is the action of trypsin? It breaks those bonds in which carbonyl group is contributed by lysine and the arginine. So here we are having the breakages. So simultaneously, if both are acting, then it will result into the breakage here. So one polypeptide, here another, then here will be the next, and here we will have the next breakage. Okay, so here is also one. So here we are getting the breakdown of this polypeptide chain. So that is the consideration for the today's lecture. So in this lecture, what we have seen, the important 4 type of the bonding, that is the hydrogen bonding, ionic bonding, disulfide bonding, hydrophobic bonding and how they are stabilizing the structure. Then secondly, we have seen a small topic that is the prosthetic group and what are the examples. And lastly, we have seen the action of an enzyme on the polypeptide chain. In this, we have seen a proteolytic enzyme that is a trypsin and chemotrypsin which break down the polypeptide chain. So as far as the examination and understanding of protein is considered, four type of the bonding and action of enzyme is one of the important topic. So here we are ending the topic protein with these all three lectures which cover the important things which are included in the syllabus for the protein and QI level. So lastly, thank you for watching this video.